Alright, so you can check your answer as we go along and add on wherever notes that is necessary. Okay, for the first question, where we are going to select which statements about both the mitochondria and the chloroplast are correct. Alright, so the first one, they contain ATS ribosome. Now that is actually incorrect. Right, instead of ATS ribosome, they only contain the smaller version of the ribosome, which is the 70S ribosome. Right, the ATS ribosome is what we have, right? Ribosomes, what we have, which is attached on our RER, right? So that is the typical size of our ribosomes, which is ATS, which is a bigger version of the ribosome, which are made out of rRNA, right? And then just a bit of revision, remember that ribosome is the site for protein synthesis. Right. The next point is that they contain circular DNA molecule. That is indeed correct. Okay, so we're gonna highlight that one. It's definitely what you need to know. And they also produce ATP. Of course, right? Mitochondria produces ATP in the process of what known as the respiration. So aerobic respiration. You are going to learn this into more detail when you go into A2 level, whereas the chloroplast produces ATP in the process via photosynthesis. Again, you are going to learn this into more details when you enter A2 level next year. So the correct answer here would be D. Which structure is only found in a typical eukaryotic animal cell? Now, a little bit of a definition. Okay, what is a eukaryotic cell? Now, eukaryotic is basically any cell with a membrane-bound or nucleus, right? And a membrane-bound. They also have membrane-bound organelles. Okay, in our syllabus, we also need to know example of prokaryotes, right? So remember that prokaryotes, right? Okay, which is the opposite of the definition of a eukaryote, right? So prokaryotic cell, okay, is any cell that lacks membrane-bound nucleus. Oh, sorry. It also lacks membrane-bound organelles. Right, I'm sure you would remember what are the examples of membrane-bound organelles. Okay, for example, there are examples of organelles which have double membrane, for example, the mitochondria and the chloroplast, right? So for example, mitochondria, chloroplast, that's a double membrane, of course, including the nucleus itself. But we also have, for example, the lysosomes, okay, the RER, the SER, the Golgi apparatus, all right? Whereas ribosomes, they do not have a membrane. Okay, but what we do know is that ribosomes, especially the ATS ribosomes that we have, it has a large subunit and a small subunit. Okay, so that's an example that we can you can add on into your notes. All right, so which structure is only found in a typical eukaryotic animal cell, right? In this case, okay, for the prokaryotic cell, the example that we need to know is bacterium, a typical bacterium. Right, and in our syllabus, we need to know that a typical bacterium must always have, number one, cell wall. Oops. And that cell wall is made up of peptidoglycan. Right? So the second one, it will always have a cytoplasm. And then it will also always have the cell surface membrane. Okay, so anything else, right, it will not be present. Right, so in a typical eukaryotic cell, only found, okay, because we can also find, number one, cell surface membrane. We can also find it in the bacterium here, right? Centriole is found in the animal cell, especially only in animal. Cells. Remember, animals are eukaryotic, right? Golgi body also. Now, Golgi body is an example of membrane-bound organelles. Whereas ribosomes, both, okay, prokaryotic cell also will have ribosome. However, they might be slightly different. Okay. In this case, the typical correct answer here would be C, which is the Golgi body. Right? Just to take note that ribosome is also present in prokaryotes because ribosomes are not an example of a membrane-bound organelle, right? So this one is a not membrane-bound. Because instead of membrane-bound, what we know, like I mentioned earlier, it has a large subunit and a small subunit. But of course, the difference is that the ribosomes in prokaryotic cell tend to be much smaller. All right, so that's why C is the best answer. All right, in order to do this next question, all right, so we are required to calculate the actual width of the organelle labeled A as shown by the line X to Y. All right, X to Y. All right, so this is the transmission electron micrograph of a part of an animal cell. All right, so I don't know if you have noticed, right, by the way, that this is the structure of a mitochondria. All right, it's a huge, nice image of the mitochondrion here. Okay, just a little bit of revision for everybody. You can always take note as well, right? It's gonna be useful to take notes anyway, and why not? Okay, right. So this is the structure of a mitochondrion. Right? We know that the mitochondria is an example of an organelle that has a double membrane. Okay. Right? These finger-like projections that you can see, that is the cristae, which is the infolding of the inner membrane. And then the fluid filling the structure of the mitochondria, that's called the matrix. Okay, and the cristae is where you can find a lot of enzyme that is needed to make ATP. It increases the surface area, right? Increases the surface area for enzymes and proteins that is needed to make ATP. Production or ATP synthesis, okay? Right. Okay, moving on. You can see that B is pointing to the next particular structure. Okay, this is basically, you can notice that it is a series of membrane, right? And I don't know if you can manage to see this, but if you zoom in a little bit, we can see these dots structure. So those are ribosomes, which means that this structure B is actually the rough endoplasmic reticulum. Now, I keep saying to my students that they should be labeling it with capital letters. Just for the sake here, I'm just going to change it. So if this was like a biological drawing, in case, let's practice labeling with capital letters. All right, so how do we go about with this question? So the first thing that we might need to do is they want us to state the formula. Now, in our syllabus, okay, you're actually allowed to simply state the formula. Now, I do encourage the student to actually use the triangle because I'm a visual learner as well, right? It helps me a lot. So magnification is image size over actual size. Right. And if we cover 1, so we were looking for, or we want to calculate the actual width. So let's just highlight that bit. So actual width would be A is equals to I over M, magnification. Now, where do we get the numbers for I? What you would need to do in this case is you're going to be measuring the line X to Y. So you're going to be measuring this. You put your ruler here, right? Measure your ruler. Okay, now I always encourage the students to measure them in millimeters and then try to convert them into micrometers. As you can see, even here in the answer, you are required to give your answer in micrometers unit. So whatever is the measurement there, okay, you need to convert that into micrometers unit, like approximately on my piece of paper, okay, it should be around 40 millimeters length. Okay, so 40 millimeters. Okay, and then divided by the magnification. They do give us the magnification value here, which is 16,000. So put that in, 16,000. However, we're going to convert this units into micrometers first. So we just proceed that by timesing. So what we do, we multiply that with 1,000. So now you have 40,000 micrometers divided by 16,000. All right, so you can just do the cancellation, use your calculator, and you should arrive to the answer of approximately 2.5 micrometers unit. So 2.5. Now remember, in the exam, you must always have the answer here in where it should be. Even though you have a full mark here, you completed all the working, but there's no answer here, you will lose 1 mark. Okay, just be very, very careful. Now, in the exam, you're also allowed to simply write the formula of magnification using these letters. Just simply A, I over M, or M is I over A. This is also acceptable as part of your formula, so that should be all right. Okay, next it says, name the organelle A and states its role in the cell. So we've mentioned that already, that A is the mitochondria. Okay, this is a mitochondria, right? And then we know, and I do suggest that you guys write this answer as well, that so it's a mitochondria, but because it's one organelle, technically it should be named as a mitochondrion. This is for singular. If it's plural, it will be mitochondria. For plural, if you have more than one. Now the role is, this is the site for aerobic respiration, to produce ATP. Now a little bit of notes, right? Remember that ATP, right, is known as adenosine triphosphate. Adenosine triphosphate. Okay, this is the energy that all living things utilize. Okay, that's A-level answer. Now, structure B is the rough endoplasmic reticulum. Now, how do we know? Because of the features. They say that give the reason for your answer. It's basically, what are the features that allows us to identify that that is indeed the rough endoplasmic reticulum? Simply because if you look here, you can see that there is a lot of ribosomes attached on the membranes of the endoplasmic reticulum. So of this, right, all of these small structures, small dots that you can see, those are ribosomes. So simply you can write down the answer here. The reason why you name it or you identify them as the rough endoplasmic reticulum is because there are ribosomes on its membrane. Okay, a little bit of notes. Rough endoplasmic reticulum. Okay, we know that it contains transport vesicle. Not it contains, it will release transport vesicle, where what's the function is that it will transport the protein or the polypeptide which is made in the ribosome. Where is it going to be transporting it to? The Golgi apparatus or the Golgi body. Okay, you can take note of that. Now, some additional notes, and well, just to take note, I know it's repetition, but it's good to repeat sometimes. Now, remember that the features that a mitochondria would also have is that it could also have E circular DNA. This is the same as the chloroplast, and it will also have a 70S ribosome. Now, having ribosomes would imply that, right, mitochondria is able to actually undergo protein synthesis. It will also makes proteins that it would need. Okay, so you can take note there. Moving on to the next question. This is the photomicrograph of a mesophyll tissue of a dicot leaf. So you can see that this is a very nice image of a dicotyledonous leaf, right? So what is the magnification of the photomicrograph? It says. So of course, we need to remember what's the formula of magnification. So formula of magnification is M is equals to I over A. Now, where do we get that image size? Where do we get the actual size? Now, the image size is the measurement that you're going to measure the scale bar. So what you need to do is going to measure this scale bar. Okay, the measurement of the scale bar, that would be the image size. Okay, scale bar. Okay, I'm going to highlight that with the same color. That is your image size. Now, the actual size is stated underneath that scale bar, which is 5 micrometers unit. All right, 5 micrometers unit. Now here, okay, approximately, now I know that if your question or worksheet is printed in a 2-in-1, it might not be exactly the same answer because of the scaling issue, due to the printing issue. But the correct answer here, if you have this printed on a correct A4 full-size paper, it should be B. Okay, notice that the units for magnification, we always put the X, that one to indicate number of times. So remember, magnification is the number of times larger the image is from the actual size. So there you go. So there's one, just put times at the front of the number of magnification that you have calculated. Okay, the next question is the electron micrograph shows a structure that found in the cytoplasm of an animal cell. Right? Now, this image shows characteristic feature of the structure. Now, this one, I hope everybody should be able to recognize that this is acentriole. How do we know? Because a centriole contain not 1, not 2, but 9. So let's just count. It has 1, 2, 3, 4, 5, 6, 7, 8, 9. It has 9 microtubules triplets. It has 9 microtubule triplets. And if you notice the interesting features that we can also find here, I'm going to use red, hopefully you can see it better, is that the way the orientation of these triplets is almost like it's 90 degrees to one another in the next triplet. Right, so really, really interesting. So that's one of its identifying features that we should not miss, right? And hopefully you can remember this in the future too. So this is how a centriole would look like. All right, and remember that centriole is only present in an animal cell. You will not find it in the plant cell, right? Because it contains microtubules, which also means that microtubules are also only found in animal cells. Okay, and a little bit of revision. What is the function of the centriole? Okay, they are involved in cell division in the formation of the spindle fiber. Okay, especially during mitosis. and meiosis, which is during the cell division. In fact, when we actually do learn this at some point, we should clarify that this is during nuclear division as well, spindle fibers. Okay. Now, what is this cell structure? It shouldn't be a lysosome. Okay, remember lysosome has a spherical structure. Okay, definitely going to be way, way bigger than the ribosome. All right, and remember the function of the lysosome. Okay, it contains, I just put that green as, just to indicate that this is a node. So lysosome, okay, number one, it contains hydrolytic enzyme or digestive enzymes. Right? Why? Okay, to break down old, worn out organelles or the whole cell itself. Okay, and of course, I hope you also remember that ribosome. I always want the students to remember this is the site for protein synthesis. Okay, whereas vesicles are membrane sacs, okay, which carries or transports, okay, proteins. Okay, this we can find vesicles in the Golgi apparatus, okay, which is known as the Golgi vesicles, all right, as well as the RER, which is also known as the rough endoplasmic vesicles, or you can just say the transport vesicles as well, boleh juga, okay, that works as well, or RER vesicles. Okay. For the next question, we are going to look at how we are going to calculate magnification of a drawing. Now, when we want to calculate magnification through a light microscope, there's a special ruler that we can see in the microscope, which when you look at the inside the eyepiece, in the eyepiece, there's something called the eyepiece graticule. All right? Okay, if you see here, this is the eyepiece graticule that you would typically see, okay, under the microscope. We actually put this graticule inside that eyepiece, okay, the one that you use to view your specimen. So this is how the eyepiece graticule would look like. It actually has a number, but we do not know what is the scale or the values to each of these division. That's why when we do in the future, what we will do is we will do a calibration practical so that we can give a value on what is the number of each division. We will have a particular unit to this. So that's the purpose of a calibration, microscope calibration practical. All right, so we'll learn this later on. So this is an image of an eyepiece graticule. Okay, eyepiece graticule. Okay, so if you look here at this image, this is an example of how we will be able to view the cell under the microscope, and with the help of this eyepiece graticule, we can measure, in this case, the width of these cells. Okay, of course, at this moment, we won't know what is the unit of each division. That's the first thing that we will need to do, because we will need to find what is the distance between this division, and we will give a unit when we do the calibration practical later on. So take note. Number 2, because we are going to be calculating the magnification of a drawing, okay, obviously, okay, with the formula, just to recap and to remind everybody that magnification is MI over A, right? For the image size, that would be the length of our drawing. So how do we know? What do we need to measure the length of our drawing? Obviously, in this case, we would need a ruler. Okay, so number one, we will definitely need an eyepiece graticule, because the eyepiece graticule is what we will use to measure the actual length or width of whatever it is that we are drawing. In this case, the width of the cell, for example, in this case, right? So we will need this eyepiece graticule so that we can measure this, the actual width of the cell. And then we will need the ruler, right, to measure the length or the width of our drawing, right? Now, we do not need a hand lens, okay, because hand lens is just what we use to view objects. Okay, that's not going to be useful. And the next thing that we will need is called the stage micrometer scale. Now, what is that? Now, a stage micrometer scale is basically, it would look something like this. So when we're going to be doing this calibration practical, what we will put is we will put a slide which contains this scale. So this, hence why it's called the stage micrometer scale. In fact, it will look very, very small like so. All right. And then they tell us already that each division that we can see here is around 0.01 millimeters, right? So each division is 0.01 millimeter. There is 100 divisions, so 0.01 times by 100 divisions. If you notice, this one will equal to 100. There's going to be 100 lines, 100 divisions. So that is actually 1 millimeter. So that means that this entire length is only one. Oops, it's too big. I need to like make it smaller. So this is 1 millimeter. So divide that 100, that means each division would be 0.01 millimeter. Now, just to make it easy for us, remember that when we do practical, we always like to use micrometers. So what you can do is, if you want to find what each division is, so 0.01 millimeters, you times that with 1,000. So that would be equals to, right, I'm just going to use my calculator as well. 0.01 times by 1,000. That would be 10 micrometers each. Okay, so you know that each division equals to 10 micrometer. Now, bearing in mind, this is actually very, very tiny, because we're going to put that under on our stage, and we're going to view that under the microscope. When we view them under the microscope, you'll notice that it will look something similar like what you see in this image. So the bottom bit here, this would be the eyepiece graticule that you see earlier, right? Eyepiece. Okay, and then the top bit here, this is how the stage micrometer would look like. Stage micrometer. Now, even though earlier I told you already that with the eyepiece graticule, we do not know what is this each division equals to. There's no unit, we don't know yet. That's why we would need the stage micrometer, because we know that each division of the stage micrometer is equals to 0.1 millimeter or equals to 10 micrometer. And we can use this to calibrate so that we can divide each division, and we can use that to find what is the value for each division of the eyepiece graticule. Okay, I think I'll do a separate time to discuss that with you. So therefore, in this case, the correct answer for number 6 would be 1, 2, and 4, and that would be B. I don't really want to spend too much on question 7. I think you should already know by now this would be A. Okay, using the formula, I think you guys are better at that, right? Next one, the structures which are required for the formation of lysosomes and hydrolytic enzymes that they contain. Now, what do we need to know here is number 1, we know already what is the organelles that produces the lysosome. That is our Golgi apparatus or Golgi body, right? So that one is a tick. Okay, now hydrolytic enzymes that lysosome contains, enzymes, they are proteins. Okay, and where are proteins made or synthesized? It is synthesized in the ribosomes. Where are ribosomes found? They are attached on the rough endoplasmic reticulum. So we do need the rough endoplasmic reticulum. Next is, last but not least, is that this process will require mitochondria, because mitochondria produces ATP. Now, what do we need ATP for? ATP is needed for very much a lot of processes that occurs in the living thing. Okay, I think your friends have already presented this to you. Like some of the examples that we heard from your friends are that we need ATP for, say, biochemical processes. Okay, in this case, we're also making, okay, proteins, right? This will need energy. Okay, we are going to be making lysosomes, right? This will need energy. So biochemical process is one example. Now, without energy, we will not be able to do any of these processes mentioned. That's why we have to include mitochondria. That's why the correct answer is why we need all the three of the organelles. Then that's why it should be B. So 8 is also B. Last but not least, which structures contains rRNA? Just for your information, what does that stand for? rRNA stands for the term ribosomal RNA. Okay, that means it makes ribosome. And we know that there is one structure where whichever structure that contains ribosome will contain rRNA. So we know already, right, that all of the three structures that will contain ribosomes are chloroplast. Okay, but we know that in chloroplast it will be 70S. Mitochondria will also have a 70S ribosomes. Nucleus will contain ribosomes. Specifically is that it is produced in the nucleolus. All right, and therefore the correct answer is 1, 2, and 3. 4 is not available. Smooth endoplasmic reticulum does not have ribosome. It does not produce ribosome either. So the correct answer here would be B. So please take note. All right, and of course, you should, if you do not know by now, know all of the functions of whatever organelles that we have seen here. All right, okay, if you haven't, that's something that you need to do for your revision in your own time. Okay, I hope that is helpful for you guys and you're able to take some notes that is necessary. And by the end of this, you get better understanding and you can remember more on the structures and functions of the organelles.